And before getting in lecture, are there any questions right now about, I know the problem set's due on Thursday, or any other comments or concerns? Let me share my screen. Okay, so today we're going to get into moving on with the spectroscopy. We're going to get into fluorescence, which is my area of research a bit, so hopefully I can cover everything, so I can talk about this at length and much more than a lecture, but also let me know if you have questions throughout, if there's something that I skipped, since it is an area that I know pretty well. And before talking about fluorescence, I just want to give you guys a status that I'm going to check the COVID numbers and dashboards tonight about what the status is on campus. I put up this XKCD comic. If you guys haven't seen it, it is actually about University of Illinois, Urbana-Champaign, where I did my postdoc, where these two physicists mentioned here that came up with a model. They're actually biophysicists, and their model did not do so well of like how to have the number of cases on campus. So as a biophysicist, I'm not going to have a big ego here and say that I know how the numbers are going to go. But I do think possibly for next week on Thursday, I'm going to talk about our class next Tuesday is going to be the first lab visit to BioBox. And we're going to have breakout groups for that. So with that sign up, I might, depending on weather, might have an option for people, if there are people who want to meet in person outside for that breakout group. If there's enough interest, that might be the first like in-person meeting that we can have. So I am thinking about this. We'll see what the numbers are and what options there are for maybe some in-person meetups or in-person in class. So still, I guess, TBD. Okay, shifting back to fluorescence, we're going to be talking about, this is a different type of spectroscopy from the absorbance that we've been mentioning. So we're going to be focusing on fluorescence, or more broadly, luminescence, which we'll define what those are. Important in fluorescence is representing the energy transitions and knowing the time scales of different processes in fluorescence. And this is called the Jablonski diagram. So how do you draw those and how do you represent what's taking place? We'll also talk about not just the basic fluorescence instrumentation of just looking at the emitted photons, but also some more advanced methods that measure distance. So this will be fluorescence, resonance, energy transfer we'll go over, and also rotation. So this would be anisotropy. And then of course, we're gonna go over the instrumentation that's involved and the biophysics applications. And we'll go over the biophysics applications more briefly today because we will be talking about fluorescence in terms of imaging, in terms of looking at diffusion, and for future lectures as well. So today, the really key thing is what's going on behind these luminescence processes and the energies as well. So the instrument that we'll be going over today is more just bulk ensemble fluorescence. So at the ensemble scale. And here, again, our source, this is spectroscopy, so we're still using photons. It's mostly going to be focused in the visible range, but it can go from the UV all the way to the IR, depending on what you're looking at. For our sample, for our biophysical sample, you can look at intrinsic fluorescence. So that means the biomolecule itself has something in it that is fluorescent. Or you can have something that you can put a label on the outside. So we will be going over how you label your molecules if it doesn't have something fluorescent. In between here, also we'll have a wavelength selector. We'll go over this in more detail. And then the detector, the important thing is we're looking at emitted photons here. We're not looking at absorbed photons, and this will be a photo detector. So with fluorescence, what we're talking about is different from what we've been talking about the past 2 lectures, that we're focused on absorbance. Was the last 2 lectures. So with absorbance, we were looking at, we start with some intensity of light. There's a sample, it'll absorb some of that light, but what we're detecting is the photons that pass through. So this intensity after the sample, those photons are the same as the photons that were coming over here. Fluorescence is different. Fluorescence is a form of luminescence. We'll talk about both forms of luminescence. There's fluorescence and there's phosphorescence, but luminescence overall is that you have emission of new photons. From your sample. So they absorb some light, it interacts with your sample, and then new photons are emitted from that. So some general, so there's fluorescence and phosphorescence. Where we'll go over the energies of this. Fluorescence is more common, it's faster, and it's from a singlet state. We'll go over that on the next slide, or in 2 slides. And phosphorescence is slower, and it's from the triplet state. We'll talk about that. So those are the 2 forms of luminescence. Some key things with fluorescence that are characteristic of it is it always has a Stokes shift, which means you have a lower energy photon than your excitation. So you can see over here, these are some fluorophores. They're emitting different colors, but they're all being excited. If you notice, this is like a UV lamp here. So that's a bluer excitation compared to the emission here is redder than the excitation. So you can always say that it has a red shift to the lower energy of the electromagnetic spectrum. We're going to use the term fluorophores or dyes are fluorescent molecules, and these are typically aromatic, meaning that they have this conjugated structure of alternating double and single bonds. So you can see here some typical fluorescent dyes that are used, where all of them have, you can see these pyrene or benzene type groups. And based on the length of those and the groups attached to it can shift the color redder or bluer based on like where the electrons are being shifted. And then this is commonly used in the visible range, like I mentioned, but it can extend to the UV or the IR, the near IR. We're developing dyes that work in the near IR is actually like a hot topic of research right now, since you can have less phototoxicity or damage to your samples when you're trying to look in biological samples. But fluorescence is actually a very key technique in biophysics. I would say it's almost the most commonly used because it's very versatile. You can use it at different time scales. The fact that you can label things with dyes means it has high specificity. It has very high signals compared to other methods. And the fact that it can be used in the visible region as humans, since we can see visible light, it's very nice to use that, and is why it's a routine tool for all these properties. I did want to talk a little bit more about the fluorophores and the aromatic structure of the dyes. That I've brought this up with the absorbance as well, and that the energies for these conjugated molecules fall within the visible range. And I really like the example of and the application of how quantum mechanics and like using the equation for particle in a box can be applied to understanding why fluorophores absorb in the visible range. So I have this example of fluorophore here. This is more in the UV range, so I wouldn't say you see it used as commonly in biophysics, but just as an example that has a nice simple structure, I have this 1,4-biphenyl butadiene. And if you remember from quantum mechanics, you can approximate particle in a box with the equation here, where the energy level is Planck's constant squared times the quantum number squared here over 8 times the mass of your particle times the length of the box squared. So in fluorescence, you're absorbing a photon from your highest occupied molecular orbital to your lowest unoccupied molecular orbital. So you're looking at the energy difference between two energy levels. So what this would be your quantum number state of your excited state minus the quantum number of your ground state squared all over 8 ml squared. So with this molecular structure, we can approximate the length of a carbon-carbon bond as about 0.15 nanometers. So a rough approximation of the length of our box here would be 1 nanometer or so. And then knowing that with these conjugated molecules, it's the pi electrons that are participating in, I'm drawing their molecular orbitals here, that are participating in this transition. So if we count the number of pi electrons in between these phenyl rings here, we have, I guess, 2, 4, 6, 8 electrons. So with our molecular orbitals, our quantum number of the ground state would be 4, and the excited state would be 5. And if you plug 1 nanometer in here for the length, and these quantum numbers in here, and you carry out this calculation, what you get is your energy transition is 3.38 electron volts, which if you convert that to nanometers, it's 367 nanometers. Which if we compare that to our spectrum, this is the fluorescent spectrum, this is the absorbent spectrum. You can see if we kept 367 is around here. So it's a very simple calculation and approximation, and you can get very close to what that fluorescence emission wavelength would be. So I just wanted to show this as a quick example of like how you can think about why conjugated molecules have these nice visible transitions, that it really can be approximated by something simple from, well, something from quantum mechanics. I won't say quantum mechanics is simple, but the simple model of the particle in a box or so. So with fluorophores, this is 1,4-biphenyl butadiene. I've also shown here where it's hexatriene, or we have octa. So we have this longer chain here. So if we use these fluorophores instead of the butadiene, what would you expect to change in your fluorescence spectrum here as you increase the length, change this here? What would change in your equation here, and what would you expect with the spectrum? You guys have any thoughts with that? In the chat, or you can speak up. Yep, so Colin's saying that you'd have, let's see here, so L would increase. Anything else would change in this equation? So Rose's guess is a smaller wavelength. Yeah, so the quantum numbers would change. Yeah, so the quantum numbers in the lake change. You can see that these are in the numerator and the denominator, but if you actually carry out these calculations, and I suggest you guys try this, but as you plug in these numbers, as the length increases, the delta E will decrease and you're gonna have a red shift. So working through this exercise, try this out with hexatriene and an octa tracing is a good exercise, and you'll see that this red shift happens. So I'm bringing this up because when you look at the structure of different fluorophores, over here, you can see that the shorter, I guess these quinine is in the bluer region versus as you shift redder, you can see here that you have this alternating bond here, that the larger your fluorophore is, the redder it's going to be. And there's some trade-offs between using, as I mentioned, working in the red range, you can have less photo damage, but if your size of your fluorophore is going to perturb your sample because it's too big, there's some trade-offs there. So Andy's asking if this only works with the 2 phenols. So this is very nice approximation because it is a simple fluorophore, but this particle in a box approximation can give you a rough number to start with. Like I mentioned, if you start to add different chemical groups here, like NH2 or carboxylic acids, those will change like where the electron distribution is based on the polarity of what you're, or if you have a hydrophobic group. So that will shift the wavelengths. So I would say this is a nice approximation. It won't work perfectly for every dye. But I always keep this in mind, like, okay, the larger the box, you know, the lower the energy is the way I think of it. Okay. So very key to working or thinking about fluorescence includes representing the energy transitions that take place. And these are done using Jablonski diagrams. So this is a way to illustrate the energy changes, the absorption, and emission that's taking place in fluorescence. So in a Jablonski diagram, you'll have your energy scale over here, and you'll represent your ground state and your excited state. And these are electronic states. Also, you'll have your vibrational energy levels as well. So this is where you start with your Jablonski diagram. First thing that happens in fluorescence is you absorb a photon, and that will excite an electron from your ground state to an excited electronic state and an excited vibrational state. So step 1 is absorption. And this typically takes place on the instantaneously, as they say. Or I guess 10 to the negative 15 seconds. So this happens so quickly that the nuclei can't move. And this is important because that if the nuclei had enough time to separate or move, that might change the energy for absorption. And this concept is called the Frank-Condon principle. That your nuclear coordinates aren't changing during the absorption process. And remember that we've been talking about absorption so far. So this is where Beer's law comes into play, is the absorption of the photon. So after photon is absorbed, you're in an excited vibrational state in the excited electronic state. And the next thing that happens is you relax down non-radiatively to the ground vibrational state in your excited electronic state. And this is called internal conversion. So when I say non-radiative, that means no photons are being emitted. So you go from excited vibration and this takes place on the order of 10 to the negative 12 seconds or picosecond or so. And some aspects of internal conversion is the fact that from here, you'll go back down to the ground state and emit your photon. So some important things here is that no matter what excitation wavelength you're using, you might be excited to like a slightly higher vibrational state, but you're always going to relax down to this ground vibrational state in the excited electronic state. So this means that your emission is independent of your excitation wavelength. And this is called Kasha's rule. And just as a side note, Kasha is my academic great-grandfather, and my academic advisor had, their dog was named Kasha. So I also think of just a huge German shepherd when I think of Kasha's rule. And the fact that this happens is also why you have this mirror image type structure between the absorption and the emission. Because the next step would be emission of a photon, and you can also go down all the way to the ground excited state, or you can go down to an excited vibrational state within the ground electronic state, and then also have internal conversion down to that ground state. But this photon that comes out, because of the mirror or the symmetry between the vibrational states in the excited electronic and the ground electronic state, that's why you get this mirror image here between the absorption spectra and the emission spectra. So the emission is called, that's the step that's called fluorescence. And the time scale of this can range quite a bit, but as a rough number, we would say on 10 nanoseconds or so, 10 to the 8th seconds. So we call this emission, or you can also call it relaxation. So absorption is a radiative process. A photon is involved. Internal conversion is non-radiative. There's no photon. And fluorescence is then again radiative. There is a photon involved. So that's a basic, if you want the most simple diagram of fluorescence, this is what it would look like. But there's also some more complications that can become involved. You can also go to the first triplet state. So if the energy, if your electron changes its spin, it can go to a triplet state. And this is called intersystem crossing. And without the spin change, it would be quantum mechanically forbidden for the electron to go there, but the spin flips, it'll go to the triplet state. And the triplet state, it can be dark. You can have no photon be involved with the relaxation base back down to the ground electronic state, or it can be radiative. And if a photon is able to be emitted from this state, this would be phosphorescent. So that's the fifth thing I have illustrated here is phosphorescent. And the time that the electron is in this dark state is quite long. It can be from milliseconds to minutes. And that's the difference between phosphorescence and fluorescence. Fluorescence is taking place in the excited electronic state, while phosphorescence is taking place in the excited triplet state. And the time scales between these are vastly different. So those are the basics that I wanted to show in the Jablonski diagram. I actually have a picture of Jablonski here, just as some interesting history tidbits. He was Polish, and he's considered the father of fluorescence, and he was actually in both world wars with Poland. And before he decided to focus and study physics as his calling, he was actually the first violinist in the Warsaw Opera for a while, and then he gave up music. And I know in our department there's a lot of, I've ran into several double majors in music and physics, so, and I think that's a common interest, or scientists are attracted to working in music as well. Okay, any questions with the Jablonski diagram so far? Okay. So with the Jablonski diagrams, you can also show some, you can also represent and talk about some important parameters of the quantum yield and the fluorescence lifetime as well. And we can draw a simplified Jablonski diagram of S0 going from the ground electronic state to an excited vibrational state, in our excited electronic state, and then going back down. So we're going to call here with the emission that there's some rate related to that called a rate constant for emission, but there's also ways to go from the excited electronic state. I'll draw this as a dashed line. That there's also non-radiative ways to exit the excited electronic state. Not every time that you go to the excited state do you emit a photon. You can also relax back down by emitting heat or have some competing processes. And then we'll also call in the excitation that we have a number of photons for excitation that we're going to call N absorbed. And then for the photons coming out, we're going to call it N emit. So simplified Jablonski diagram, these are all the parameters that we're going to use to describe quantum yield, which is an important parameter for dyes or fluorophores. And it's just how many photons do you get out for the number of photons that you put in. So I guess I can write this as, I could write it out in words, but it's just easy to write in the equation of the number of emitted photons over the number of absorbed photons. You can also write this in terms of the rates as well, where you can say it will be the rate of emitted photons over the total rate of non-radiative photons plus the emitted photons. So because I guess the number of emitted photons plus, yeah, and we'll put that in terms. So, and this summation is because there's multiple non-radiative processes that can take place. So I'm just, in the Jablonski diagram, I'm just writing this simply as like just one number here. So with this ratio that I show here, what are the values or possible ranges that you can get for the quantum yield? You guys can write in the chat or speak up. Yep. So Bill says you can go from 0 to 1 because it is a ratio. You can't have more photons emitted than what you absorb. And what would be the desirable value that you would want when you're doing fluorescence? Yeah, a value of 1. So 100%, the perfect fluorophore, you would get 100 photons out for 100 photons in. I would say typical ranges are more around 0.3 to 0.8. You can get closer to 1 with very optimized fluorophores, like rhodamine or alexafluorophores. But if you're working with fluorescent proteins, we'll talk about these later, you might be more around 0.4, 0.5. But this is an important value for when you're selecting different dyes. Another important parameter is the lifetime. So that's how long your dye is in an excited state. So on the previous slide, I said 10 to the -8th. This is more, I would say 0.1 to 100 nanoseconds is the broader range for that. And this lifetime, it follows an exponential decay typically, and you can get this out from 1 over the rate, which would be 1 over, again, the sum of your non-radiative processes plus your emission rate. So this is one way to express it in terms of the rate. But also if you're thinking of a population of dyes, you can also express this in terms of the number of excited molecules. So this would be the change in number of excited molecules versus time is what you would be monitoring, where N asterisk or N star times K would be the number of excited molecules after some time T with a pulse of excitation. So this is how time resolved measurements will be done, which we'll talk about later. So you can express the number of excited molecules over time with this expression. Where again, this follows an exponential, where this N naught star is the number of excited molecules at the start, like right when the pulse happens at time equals 0. So just a simple exponential decay can approximate this. So this will depend on the environment and the dye that you're using. So measuring the exponential decay here, measuring the rate can tell you about the environment of that dye. And it's also worth knowing that here, when I'm expressing K, that's again the sum of non-radiative plus the emission. And this would be like your real rate, but there's also something called the intrinsic lifetime. And this is very idealized, and this would be if non-radiative processes didn't happen. So this would be if it's perfect. So knowing the difference when you're looking at these rates, make sure that you know if it's the real rate or the perfect rate, because you can get the intrinsic rate versus the actual. Those are all related based on the quantum yield. You can work with these equations to get just the intrinsic rate versus the actual. So these are some important parameters for an individual dye. Also, once you have your dye or your fluorophore in your environment, it's also important to know what can affect that fluorophore and actually decrease the fluorescence. So there's two processes by which fluorescence decreases, or I guess three that I'll talk about. Quenching is when you have something in your solution that increases non-radiative mechanisms, or prevents excitation. So with non-radiative mechanisms, this is typically done by something coming into contact with your fluorophore. So this is called collisional quenching. So something collides with your fluorophore and causes it to go to a lower energy state without emitting a photon. So this is due to diffusion. And it can be characterized if you see a concentration dependence. So a way to describe Stern-Bolmer, or a way to describe collisional quenching is with the Stern-Bolmer equation, where do you see a change in fluorescence as you increase the amount of quencher? Is a way to answer, do you have collisional quenching? So the Stern-Bolmer equation is, how does your initial fluorescence compare to your fluorescence in the presence of a quencher change? And this would be described by 1 times the equilibrium constant of the fluorescence in presence of the quencher times the quencher concentration, which would then be related to the rate of quenching times the time that you're measuring it at times the quencher concentration. So this equation is used to, and varying the quencher concentration is a way to determine if you have collisional quenching. If you don't see a change with quencher concentration, you might have static quenching. This would also change with like temperature or viscosity of your solution, is a way to test if you have collisional quenching, because it depends on diffusion. So static quenching would instead be, you would form a dark complex. That prevents excitation. So that means you're always in the ground electronic state. So this would have a concentration dependence as well with the quencher, but it would not follow this equation. And it wouldn't have a temperature or viscosity dependence. So those are the two different forms of quenching. You could also see decreases in fluorescence due to something called photobleaching. And this is damage to the fluorophore that makes it, that are irreversible, and puts them in a permanent dark state. So this can be when the molecule's in an excited state, it can interact with like triplet oxygen and be oxidized and have some irreversible chemical change to the fluorophore. So if you're doing ensemble fluorescence measurements, how you would see this is intensity of the fluorophore versus time. You'll see an exponential decay. Photobleaching takes place with an exponential. And if you increase the excitation power, that should decay faster. We're also going to be talking about single molecule measurements and how you would see intensity versus time for a single dye. You would see it just turn off completely. It's completely damaged, you can't be excited again. With single molecule measurements, you also might see photoblinking. And photoblinking is a reversible dark state. And this is when the fluorophore is, when the excited electron is in a triplet state. And phosphorescence is impossible. So if you did a single molecule measurement and you saw decreases in intensity that then would turn back on, that would be photoblinking. So this is the bleach. This is the blink. And the time between these would be on the same time scale that we mentioned for phosphorescence. That would be around milliseconds or seconds or so. If you're doing ensemble fluorescence, photoblinking, since you're looking at many molecules at once, you wouldn't see this photoblinking process. This only shows up with single molecules. Okay. So those are important terms that you'll see brought up in fluorescence and ways that we quantify the performance of dyes, how their fluorescence changes, and the like. But how do we actually look at molecules? What is actually fluorescent within a molecule? So we'll talk specifically about what's fluorescent in proteins and nucleic acids and the like. But typically, if we're working in the visible range, you have to connect a dye to your molecule. And this is what we call conjugation. And over here there's a whole book from Thermo Fisher that goes over what type of connections you can make. But I will say the most common ones are doing some covalent chemistry to thiols within molecules. So here you can see this R1 would be your dye, and it would be terminated with this malamide group. And then that would react with a thiol, where here this would be on a cysteine residue in a protein, or you would have a nucleic acid that would have a special termination with a thiol, and then you can form this covalent bond here. So I would say this is there's not many thiol groups in proteins that are alone. They usually form disulfide bridges. So if you have a free cysteine residue, this is a nice way to connect a dye. Less common or still used, but less specific. So thiols are very specific. You can also attach to carboxylic acids or amine groups, since we have those on specific amino acids, and also at the terminus of a protein. You have the N terminus or the C terminus. So here you can see this would be, again, your dye connected to this, I can never say this one, this ester group here, and it can react with an amine, and you can form this carboxymide connection here, covalent bond. So this is less specific, but you always have COOH or NH2 around. So that's for proteins. If you think about sugars, there's always carboxylic acids on the sugar groups at the terminus of nucleic acids. But you might not know exactly where your dye goes. You have to be very controlled. And then finally, you can use some engineering and put some click groups. So here, this is an example of a nucleic acid where you can see that there's this alkene group. And if you have a dye here, this is called a click group here, and these guys will react, and here's your dye. So if you're able to engineer in these triple carbon bonds into your molecule or a clickable group, these aren't traditionally like native in biophysics, but if you can engineer them in, this is very specific, but it's not natural, not naturally occurring. It's always important when you're adding a fluorophore or a dye to your biomolecule, you have to think, where is it? How large is it? You have to think about the charge or the hydrophobicity. Is it changing? Is it perturbing your biomolecule at all? I mean, this is a question I get all the time whenever I submit a paper. Oh, well, you're putting a tag on it. How is that influencing what's taking place with the biomolecule? So you have to be very strategic. And then finally, I just want to mention quickly that there's some other labels other than these small organic conjugated fluorophores that you can also use quantum dots or nanoparticles, where these have benefits that they're not going to photo bleach at all, but they are larger, or quantum dots are typically toxic, made of cadmium and selenide and the like. You can also have chelation. So that's inorganic chemistry where like some ion will come in and coordinate with your molecule, and that can change the structure, and that can have a turn on fluorescence. So this is used for like looking at calcium ions in neurons or magnesium, or looking for specific ions that participate in biology. Also, the bottom of the periodic table periodically shows up in biophysics as well, with the lanthanides and the actinides, that they have phosphorescence and they have long lifetimes. So they're used in some very specialized cases. And those long lifetimes can avoid photo bleaching and the like. And then also there's special dyes called flash and reash that you may come across, that these are dyes that are cell membrane permeable. So if you're trying to label something within the cell, it has to be able to get through the cell membrane, and these are commercially on the market. So in addition to these other types of labels, commonly seen in biophysics are fluorescent proteins, such as green fluorescent protein. That was discovered in jellyfish in the 1970s, and it has a really interesting story behind it. The guy who discovered it was just trying to like discard samples and came in to the lab to an old sample the next day and saw that was like glowing, that one of those accidental discoveries that transformed a whole field. So there's lots of different variations of green fluorescent protein. You can see here it has this beta barrel structure shown down here. And the special thing with green fluorescent protein is that in the center of the barrel, there's 3 different amino acids or residues that can coordinate, and you can see that they can cyclize and be oxidized, that then it forms this green fluorescence. So the important thing with the beta barrel is that it brings these 3 residues, these 3 amino acids, into an orientation that it can then form this chromophore here. And if you change these different amino acids, that's how you get the different colors that you see here, where they all have, see some fruit names here, yellow fluorescent protein, cyan fluorescent protein, and there's a huge library, and people are developing new ones all the time that have special properties with how you can photo switch them, what wavelengths. And the nice thing is you can express these in cells, and they can be attached to your molecule, your protein of interest. So then you can get these very nice images where you can have a bunch of different plasmids that then change, like where this is cyan in the mitochondria or yellow in the Golgi, so you can label specific proteins at different times. That's why I choose. Okay, so we know the energetics and the time scales and numbers behind fluorescence, how we have fluorescent molecules. So how, if we have our sample, how will we measure it? And I want to go over the instrumentation quickly because it does have a lot of common similarities between the instrumentation we've been talking about. So if we start with our source, we typically have a xenon or a mercury lamp because it has to have high intensity, or laser lines in the visible region. So the important thing is having this high intensity. And I would say more historically, lamps, more recently laser lines, just based on the cost. You would then have a lens to focus this light if you're using a lamp. And then you would have your monochromator, either prism or grating. So this will select your wavelengths, spread out the different colors of light, and you have a slit to then select the specific wavelength that you want to use. So you have a small section of wavelengths or small colors that are then infringing on your sample. We're talking about ensemble fluorescence, so this is typically done in a cuvette. The cuvette is typically silica or quartz, and it's important that you don't have, it's a high quality cuvette, so you have reduced scattering. So if we were doing UV vis, we'd have the detector right after the sample, but we're trying to look at emitted photons. And we don't want the light that's passing through the sample from our source to be detected. So instead, we put our detector at 90 degrees. Because the emitted photons are going to be going in every direction, while the excitation photons are going to pass straight through our sample. So the emitted photons we detect at 90 degrees. Again, you have another prism or monochromator and a lens, and you'll detect that on your detector, which is typically a photomultiplier tube or a CCD detector. And then of course that's connected to your computer, and you have your analog to digital conversion over here. So I'd say similar instrumentation to what we've been discussing. The important thing is that it's set up at 90 degrees, and you typically have wavelength selection on your excitation path and your detection path. So with the instrumentation, you can, the most common instrumentation that you would use would be steady state fluorescence. And this would mean that you would have continuous excitation, and then hence emission of photons. So you can also call this time averaged fluorescence, and if you find a fluorimeter, this is probably what you're going to see. So typically, the output from steady state fluorescence measurements is you plot intensity versus wavelength, and you get a spectrum of your fluorescence emission. If you did intensity versus time, what you would see is just like a straight line, and this would be like at wavelength 1. You can say at the peak wavelength over here, be wavelength 2. But you would see a pretty static, steady state emission. If you're looking at photo bleaching, you would see a decrease or the like, but this would be the most common measurement in a simple situation. More specialized would be time resolved fluorescence spectroscopy, and this would use a pulsed excitation, and typically a laser excitation. And this would be doing measurements in the nanosecond time scale, looking at the changes, how I mentioned, number of excited molecules over time. This is how you would be measuring with a time resolve setup. So you can directly measure the lifetime. And you can detect how the lifetime changes based on the environment. So your typical measurement would be intensity versus time. You'd see that exponential decay, where your 1/E value would be your lifetime. where this would be on order of nanoseconds. Time resolved measurements are on expensive instrumentation though. So if these are typically home built, or if you have a commercial instrument, it'd be something by like picoquant, and this would be running in the upper tens of thousands to like 100K or so. So that's less commonly used, but it is a measurement to be aware of. So we have steady state measurements, time resolved measurements, and getting into some of the more specialized ways that you can use fluorescence include fluorescence resonance energy transfer. I have listed here, this is also reserved, abbreviated as FRET, and it can also be Forster, who is the guy in the 1800s who discovered this phenomenon. And what happens in fluorescence resonance energy transfer is you have two dye molecules. You have a donor and acceptor. The donor is bluer, the acceptor is redder. And what happens, we draw our Jablonski diagram, is you excite from your donor molecule, internally converts, so it's here, in the excited electronic state in the donor. And what happens is you have, let me redraw this. Your acceptor molecule, which is redder, so it has a smaller energy difference between the ground and the excited state, that you have the energy transfer from the excited electronic state of the donor to the excited, I'm going to drew this arrow off, sorry, to the excited electronic state of the acceptor. This is non-radiative. It has to do with dipole coupling between the donor and the acceptor, the orientation of their electric dipoles. And from here, you can relax down to the ground electronic state of the acceptor. So you can see that you would have this redder photon being emitted. The excitation is taking place in the donor, the emission is taking place in the acceptor. And the key thing with FRET is this depends greatly on the distance between the donor and the acceptor. So you can see this fall off here is quite drastic. It follows an equation of 1 over 1 plus the distance times the Forster distance raised to the 6th power. So the fact that this is to the 6th power has this sharp drop off. And this R naught typically is on the order of, let's say, 2 to 10 nanometers. So you get curves that look something similar to this. So as the donor moves further away, you see most of the light being emitted from the donor, but as it moves closer, you see more emission from the acceptor. So this is called the FRET efficiency. And the fact that these length scales are on the same length scales as biomolecules it allows you to probe conformational changes in proteins, in nucleic acids, and it's called a nanoscale ruler. So this is just more of a static picture of that animation. So this can be done with steady state measurements, where if you look at the donor to the acceptor ratio, or typically the FRET efficiency can also be reported as the intensity over of the acceptor over the total intensity of the acceptor and the donor is another way to write this as well. So by looking at the peak emission, if you take a steady state spectrum, this is your wavelength, you can take these intensities and figure out the conformation. So if you saw something like this. This would mean that the acceptor and donor are closer together versus further away with these type of example sketches. Quickly, since I knew I was probably gonna have a lot to say about fluorescence, another advanced technique is looking at polarization or the rotation of molecules. So fluorophores with their conjugated structures that I showed, we have something like this. They preferentially absorb light along the orientation of their dipole here. So based on the conjugation. So if you use polarized excitation, you'll only, and you have a random distribution of your dyes, the only ones that will be excited are the ones that are aligned with that polarization. So just the green shown here. And then if you probe or detect these molecules emission over time, you can get how fast they're rotating. So typically in anisotropy, you would do measurements of looking at two different polarizations of the excitation light, parallel and perpendicular to one another. And by taking the ratio of the intensity here, you can get the orientation of your molecule. And you can look at this over time if you use a pulsed or time resolved measurement. You can look at the rotational, the rotation at the initial pulse and look at this versus time. With this equation, where this will be your rotational constant. Which for biomolecules is on the order of tens of nanoseconds. So you can see how these are reorienting themselves. This is typically used, especially in membrane environments, where rotation will be slower. If you were just doing dye alone, it would be too fast to be able to be resolved. So this is looking at rotations. So getting into some of the applications in biophysics, I mean, fluorescence is used so much. We're going to be talking about imaging. We're going to be talking about diffusion in lots of different ways. For proteins, you can use intrinsic fluorescence of tryptophan and tyrosine residues. And that's what I have shown here for a protein. Tryptophan is very sensitive to the environment. That in this plot here, you can see this is W in a folded protein. And then as it unfolds at a higher temperature, you can see here, this is at 80 degrees C, that you see a shift in the wavelength. And that's because this is in a hydrophobic environment in the center of the protein, while when it unfolds, it's then exposed to the solvent. So you can use it to probe unfolding of proteins, or you can even see here, the black curve versus the green curve. This is in the presence of a polymer. And you can see that the polymer is slightly quenching the fluorescence, and we're seeing a shift, so it can tell you very clearly if there's an environmental change. In nucleic acids with DNA or nucleic acids, you can use it for sequencing. So here you can see that this curve, I can follow it. You have a single strand of DNA. You can then introduce complementary strands that pair up and have fluorescent dyes on them. And you can do sequencing of DNA by sending in different probe sequences here. That's a way that DNA sequencing is performed. Fluorescence can be used for, again, it's very specific based on the tagging. So you can use it for detection, sensing. Looking at conformational changes with FRET, We'll talk about, in future lectures, we'll talk about diffusion. We'll talk about imaging. There's just a lot of different ways that it can be used, and we're really going to go into details in future lectures as well. And then last, I just want to mention again, as I end up every lecture with some practical use, that I'm going to be talking about steady state fluorescence. That here's an example of a carry allegiant spectrometer, that it can be pretty small benchtop to handheld devices. Depending on what wavelengths you're using, the ranges, that's what's going to change the pricing. There's 96 plate readers, or just a cuvette. And the prices range for like ones of thousands to tens of thousands for steady state. And I already mentioned like the time resolve will be even pricier. It's really important for fluorimetry that you calculate carefully. I can't spell today. That the intensity that you get out is really dependent on the filters, your source, your detector. Unlike UV vis, where you're looking at a ratio of incident photons to excitation photons to detected photons, here your intensity, typically they report in arbitrary units, but if you want the absolute number of photons, it depends on all of these. Since you're not looking at a ratio, you're looking at the absolute number of photons. It's also important to know that you can have inner filter effects. Be careful that your sample itself can change intensity values. And what I mean by this is when you have a cuvette, you're filling it with your sample. If it's a big cuvette, the light that you send in, it'll interact more with the molecules that are in the front. It'll absorb some photons, and as it passes through, the intensity decreases. Because these photons are being absorbed, there's less available further into the cuvette if I have distance here. So this is called primary inner filter effect, but you also have secondary, where you have your molecules in the front will be emitting photons in every direction. And further back in your cuvette, these molecules can then be excited by those photons or interact with those photons. So this is called secondary filter effect. And that cartoon's not very good, so I'll write it out. Fluorophores in back, absorb photons emitted in front. And then the last thing I want to mention is autofluorescence, which I know some of you guys are familiar with. And that's from the fact that your sample can have intrinsic fluorescence. That's not what you labeled. So you can see here in this microscopy image that they're interested in looking at GFP that's being expressed in actin in the cell, but cells without those labels or the surrounding environment has fluorescence itself. So it's very important to take controls and correct for this. And this also happens in the bluer end of the spectrum. So that's another motivation for using brighter light as well. So I know I'm running up to the edge of the time. This is the Bible of fluorescence that I uploaded in the resources. I would highly recommend chapter 2 or chapter 1 just as an overview, and chapter 2 if you want to get into the specifics of the instrumentation. And then I've Lemke, the biophysics tools, has these chapters cover fluorescence in kind of a disjointed way of mainly focusing on microscopy, which we'll talk about later. So with that, right at 12:45, but you guys have any questions with all that content? If not, thanks for coming to the lecture, and I'll see you guys on Thursday.